This lecture focuses on biological foundations of human development. First, let's think about some false dichotomies. The mind and body were considered two separate things. This has been called Cartesian dualism. But the mind and body are intricately connected. So the mind-body is a false dichotomy. Nature-nurture, which is most important in influencing human development and human behavior. Nature, the way we're born, or nurture, the way we're cared for. It's a false dichotomy because both are important, and the two are intertwined. So much so that after a few years, it's impossible to tell whether outcomes in early childhood are due to nature or nurture. It's almost always both. Can you think of other false dichotomies that affect how we think about human development and human behavior? What about gender? What about sexual orientation? We'll talk more about those issues in a few weeks. So let's look at genetics and genetic influences on human development and human behavior. Old studies looked at whether developmental outcomes were more closely associated with nature, that is genes, or nurture, that is environmental influences. So the initial round of genetic studies used twins, and they looked at twins raised together, raised apart, identical twins, fraternal twins, and the answer in these studies, did nature or nurture matter, was almost always both. Nature and nurture matter. So we've sort of stopped asking that question, which is it? It's both. And newer studies look at how much nature and nurture interact to produce outcomes. So how the environment, quote, gets under the skin and affects development is an important line of work. We have clear evidence of gene and environment interactions. So, for example, the diathesis stress model suggests that an inherited predisposition for a certain condition or disorder has to be activated by an environmental risk factor, such as stress, in order to actually produce that condition. So if we inherit a predisposition, it may or may not be activated. And there are a variety of other epigenetic processes. In this context, the term epigenetic refers to environmental effects on gene expression. Gene-environment interactions are important, and the Rudder article talks about many different ways in which this happens. You don't need to know this in depth, but do know that genetics provides a template and a timetable for development, and experience shapes development. So the genetic blueprint is just a rough draft that can be altered by the prenatal environment, by exposure to risk and protective factors, by trauma, and so forth. The genetic code has to be activated, and to do this, transcription factors or molecules control gene expression. They control whether genes are activated, turned on, or turned off. This is an illustration, but you don't need to know this in great detail. There have been important critiques of recent human genome studies. So the Human Genome Project, over the course of a decade, involved mapping, that is identifying and naming, all of the genes on the human chromosomes, on all 26 pairs of chromosomes. And then scientists tried to identify which genes were associated with which conditions or diseases or disorders. So they looked at thousands, hundreds of thousands of correlations between genes and conditions. Think about this as a giant matrix where you have, let's say, genes represented by rows and conditions represented in columns. A huge matrix of possible relations. When you look at that many correlations, you have a problem called fishing. You'll also think about this in your research classes as type 1 error. If you look at a lot of correlations, you're going to identify many spurious, that is false, associations between specific genes and diseases and conditions. And indeed, that is what happened. So in the Human Genome Project, this attempt to find correlations between genes and disorders identified many so-called discoveries, relationships, that could not later be replicated. So for a while we were seeing a lot of headlines in the New York Times and other papers, the gene for Alzheimer's has been discovered. And then another lab simply couldn't replicate the discovery. And that went on for a long time until scientists realized they were going about this all wrong. Reanalysis of the genome association data shows that gene-condition associations are modest, fairly low, because nurture matters also. And there are fewer associations than we previously thought, and those associations are complicated. It's not the case that there's a single gene that's responsible for a particular condition. It's certainly not a complex condition such as substance abuse or mental disorder. Let's turn to thinking about brain and nervous system development. We say that the brain and nervous system are experience-dependent and use-dependent. That means that experience literally affects how the brain and nervous system evolve. Our experiences shape neural pathways. And our neural pathways are affected by how often they're used. I'll say more about this in a minute. There's also a hierarchical organization of the brain and central nervous system, and the overall pattern of development here is that the brain and central nervous system develop from the bottom up. Okay, so in utero and in the first months of life, the first thing that develops is the brain stem, the sort of bottom of the brain, the spinal cord, and then the midbrain going up into the brain. And these areas regulate body functions, breathing, locomotion, and so forth. A basic architecture is established, so the anterior, posterior, meaning front and back of the brain, evolve. The middle or medial of the brain, the sides of the brain, the lateral side, the dorsal and ventral portions of the brain evolve, all in utero, and they continue to develop in the first months of life. In the first 3 years of life, the limbic system evolves in the brain. This is associated with emotional processing and regulation. So it's important to know that the fundamental structures for emotional processing and emotional regulation are established in the first 3 years of life. Also, the cortical areas, the outside portions of the brain that are associated with cognitive and executive functions, develop in the early years. But the brain continues to develop through adolescence and even early adulthood. The neocortex and prefrontal lobes develop last, and these are associated with higher cognitive functions, such as math and hypothesis testing. And here, layers of neurons are developed from the inside out. So again, the basic pattern of brain development is bottom up, inside out. Here are a couple of illustrations of portions of the brain showing that bottom brain stem, the midbrain, and the frontal and other lobes. You don't need to memorize these terms. Here's some more terms that you don't need to memorize, but it's nice to know that you have a little glossary if you need them. So how is the brain wired? Here's the first slide of several. This is how it happens. It happens through processes called synaptogenesis and myelination. Now, a synapse is the gap between two neurons in which neurochemicals are placed. So one neuron releases neurochemicals, and that sets the other neuron to fire. Okay? Synapses are literally the gap in which the neurochemicals are released. So synaptogenesis means that we are creating synapses. And myelination means that we are insulating neural pathways that are used often. So how does this happen? First, in utero and in the first few years of life, there is a rapid proliferation of brain cells. Neurons are growing and glia are growing in the brain. And then the cells migrate to form layers and structures of the brain, the pictures you saw earlier in the first 6 months. Then connections are formed between cell groups around 7 months in utero to age 2. and synaptogenesis, again, the linking of neurons, we informally call it wiring, via dendritic axons, the long fibers, and neurotransmitters takes place. So we create these connections between neurons. Now, when those connections are used often, they become myelinated. And that means that fatty myelin cells surround neural circuits, multiple neurons, and synapses. So now we are basically insulating a neural pathway. This reduces static or outside interference, and it speeds transmission. But it's associated again with frequent use of a pathway. And you can almost see this happen when you watch children learning to walk. So at first, when they're taking steps, it's a very halting process as the brain is trying to tell the legs and other body parts what to do. And the more practice young children have with walking, the smoother it gets. And what's happening here is that those neural circuits that are controlling locomotion are becoming myelinated. So you can see, literally see, that the process becomes more smooth. So what happens next? In this process, there is an overproduction of synapses. We produce lots and lots of neurons and lots of synapses, and some of them don't get used. And so there's a process called pruning that happens. The synapses that are not used die off throughout childhood and adolescence. So this is the sort of use it or lose it idea, and it helps us to organize and specialize brain circuits. There are critical periods for development of some brain functions, such as vision. So, for example, if the brain is not exposed to visual stimulus in the first months or years of life, we will not learn to see. So, if the retinas are not attached and the brain doesn't get signals from the eye, attaching the retinas much later in life will not help. The brain must have visual stimulus during a critical period in order for vision to develop. Now that's very unusual. There are more common are what we call sensitive periods. So there are times when it's best to have stimulus, but if we get that stimulus later on, we can still develop the function. So one example is hearing. If children are born deaf and they don't have cochlear implants till later in life, when they get cochlear implants and can hear, the brain can process that information. Now, it's best if the brain gets auditory input early on. That will be easiest to process. So that's a sensitive period, but getting that information later does not necessarily interfere with the brain's ability to process it. So language is has a sensitive period, and this is why it's easiest for humans to learn languages in the first few years of life. Children who are exposed to 2 or 3 languages in the first couple of years can easily pick those languages up and keep them separate. And it's much harder for us to learn additional languages later on. So as the brain evolves, there's a process that we call scaffolding, and that is the sort of basic architecture of the brain develops. The basic structure, the lobes, the midbrain that controls emotional regulation are a basic structure. But this architecture can change, and we can still develop functional alternatives. So if a portion of the brain is damaged early on, it's very possible that another portion of the brain can take on those functions. The scaffolding can change. So again, we're back to these ideas of equifinality and multifinality. Let's say a little bit more about sensitive and critical periods for development. Again, the sensitive periods are when the brain is particularly responsive to certain experiences or stimuli. And a critical period is a little more serious, and this means that the presence or absence of an experience results in irreversible change. There aren't too many examples of that. I said vision is one. Now let's think about the stress response system. The stress response system is what happens when we go into flight or fight response in response to a threat. So if we are responding to a real or perceived threat, our instinctual response is to fight or flee. Here's what happens. The amygdala in the midbrain signals a threat, and the HPA system, which involves the hypothalamus, the pituitary, and the adrenal glands, release hormones which increase the amount of cortisol flowing in the bloodstream. So the amygdala says, hey, there's a problem. The HPA system releases hormones, and cortisol increases. Now, cortisol frees energy for action, but it also has some other effects. It suppresses immune functioning. Over the long haul, it can suppress physical growth. It suppresses emotional regulation and memory, both the storage and retrieval of memory. Now, that only happens when there is prolonged stress that produces anxiety and a constant state of alertness. So if we have prolonged stress, we are going to have high levels of cortisol over a long period of time, and that can negatively affect development. Specifically, it can affect affect regulation, learning, and memory. A one-time event is not going to have that impact, even early on. So there's a sensitive period for development of the HPA system, and that's early on. The other important concept here is plasticity and change. Plasticity means change, flexibility, and there is more plasticity in terms of brain functioning and brain development in the early years. The brain is more reactive to environmental stimuli in the early years. That's again why we can pick up languages more easily. Positive experiences, such as responsive caregiving, stimulation, learning experiences, support optimal brain development in the early years. So well-timed stimulation is important. It's important that we provide infants and young children with a lot of good stimulation. Infants and young children are very vulnerable to early negative caregiving, to trauma, and to malnutrition because the brain is forming so rapidly at this time. The capacity for self-regulation is more likely to be affected than cognitive development. So under negative experiences or stress, we're still likely to be able to develop ability to do math and read, but our emotional regulation is most likely to be affected. We're also better able to recover from trauma or deprivation in the early years. So early intervention is important. So one example in your readings is that kittens can regain sight that's lost due to inactivity if the activity happens early on. This underscores the importance of early attachment relationships and the importance of early stimulation and early care. It is clear that plasticity, our ability to change, our flexibility, declines over time, but some change is almost always possible. So it's never too late, but earlier intervention is always preferable to later intervention. Your readings talk about mirror neurons in relation to intersubjectivity and communication. Now, this is a controversial topic. There's some debate about whether mirror neurons are really a thing. And the idea that mirror neurons are underdeveloped in children with autism is particularly controversial. So the idea with a mirror neuron is that it's thought that when you watch somebody else experience something, the brain reacts as if that thing is happening to you. And that's used as an explanation for empathy. So I see somebody being hit, my brain acts as if I'm being hit, and therefore I have empathy with the other person. But there's actually very little empirical evidence of the activity of individual neurons. We don't see individual neurons responding. This is a theory that is not empirically supported, at least not yet. Now, we've been talking about cognitive neuroscience, and there are a lot of critiques of cognitive neuroscience. There's a lot of theory here, but it's more theory than science because of some problems in the way neuroscience proceeds. Now, earlier I talked about problems with the Human Genome Project. There are a lot of fishing for relationships between genes and conditions, and because of that fishing, scientists discovered things that weren't really true. They didn't hold up on replication. And there's the same kind of double dipping going on in cognitive neuroscience. And double dipping refers specifically to the selective use of data for identifying and studying patterns of brain activity. And we have a problem called dependency. So in the cognitive neuroscience studies tend to involve very few people, samples of 10 or 15 people, so that's few brains, and lots of images. So the imaging studies take thousands of pictures of a single brain, and each of those pictures has thousands, tens of thousands of what are called voxels. Voxels are like pixels. So in these studies of 10 or 15 people, there are hundreds of thousands of data points. And we've discovered around 2009 that many of these studies were analyzed incorrectly. The data were analyzed as hundreds of thousands of separate events, when in fact they should be analyzed as correlated events from a few brains. So the sample size should be 10, not hundreds of thousands. This led to huge mistakes in the analysis of fMRI data and other imaging studies. Here's a link to an article in The Guardian on the widespread misuse of statistics. This is particularly problematic in cognitive neuroscience. Cognitive neurosciences you can get a PhD in cognitive neuroscience without a single course in statistics. So the scientists don't understand statistical analysis. Journal editors in cognitive neuroscience don't understand it either. And we're seeing outdated methods for reviewing and synthesizing results of these studies. This is a huge problem. Here's some examples of articles on this problem. The problem with skewing brain images, circular analysis, double dipping, errors in neuroscience. It's a big problem. It's one of my favorite studies illustrating problems with current cognitive neuroscience. Here's a study called Neural Correlates of Interspecies Perspective Taking in the Postmortem Atlantic Salmon. What's this about? Here's a poster from the study. It's a real study published by Craig Bennett and his colleagues. Here's what happened. They gave a dead Atlantic salmon a mentalizing task. Now this salmon was truly dead, had been in the market, the fish market, all morning. They purchased it in the afternoon. They took it to the lab, and they put it in the fMRI machine, an imaging machine, and they gave it a classical mentalizing task, tracking the brain activity of the dead fish in the fMRI. There were 15 stimuli in this mentalizing task, and these were pictures of humans in various social situations. Each stimulus was presented for 10 seconds, and then there was a 12-second rest period between stimuli. So the fish is, quote, looking at a picture or not. And using data analysis typical in fMRI studies, the researchers, Bennett and his colleagues, found a pattern of activity in a specific region of the salmon brain. This activity appeared significantly more often during the stimulus when the pictures were being shown versus during the rest periods. Okay, so this is a statistically significant result. Was the dead salmon engaged in interspecies perspective taking? Was it looking at humans and registering something about their social interaction? And if not, how do you get these results? A significant pattern of brain activity when the pictures were being shown and no activity when they were not being shown. What's going on here? What's going on here is that they analyzed the data improperly. It's that simple. They analyzed the data as if they had hundreds of thousands of independent pieces of information, when in fact it was one brain, it was a salmon brain, and it was dead. The pattern might have come from just noise in the machine. When the data were analyzed appropriately, there is no difference in brain activity, of course not, between the stimulus and rest periods. But the purpose of this study is to show that you can find completely irrelevant patterns. If you look for patterns, you can find them. They don't necessarily mean anything. Specifically, if you don't analyze the data properly, you get results that are illegitimate. Here's some other articles along the same lines. A lot of published research findings are false because people are fishing for results and not analyzing the data properly. There's also a great deal of appeal of studies of the brain in the popular press. A lot of myths about what we think we know about the brain. Another very interesting study. Again, this might be for future reference. I'm not suggesting that you read these studies now. It's important to distinguish the mind, which is our sort of phenomenological experience of cognition and thinking and awareness, from the brain, which is the physical matter. Finally, let's talk about the cumulative nature of science in general, but also in relation to neuroscience. A lot of studies are produced in all areas. So there are a lot of studies on different aspects of brain functioning, these fMRI studies, for example. And to pull them together, to sum up what we know, we need systematic reviews of evidence that include a formal assessment of the study qualities. And the studies are not all created equal. Some are better and more reliable, more valid than others. So a systematic review of evidence includes a formal assessment of study qualities, and it includes meta-analysis, which is a statistical synthesis of data across studies. That's really important to get accurate summaries of what the evidence shows. Now, at the same time, there are narrative or unsystematic reviews of scientific evidence that are prone to error and bias. Let me give you an example. This is from Fishbane, an article published in 2007, and I want you to pay attention to the first full sentence here. Emotion processing occurs mostly in the right hemisphere, which is dominant in its growth during the first 3 years of life. The left hemisphere, responsible for linear, logical thinking, etc., develops later. Now, this is a widespread view that the right brain and left brain are doing different things. However, it's not accurate. So this is a meta-analysis of studies that looked at emotional functioning on the right or left parts of the brain. This is published in 2003, and the authors conclude they found no support for the hypothesis of overall right lateralization of emotion functioning. So that right brain, left brain hypothesis is not supported when you look at all the evidence. Yes, half of the studies find emotional functioning happening mostly on the right side of the brain, but the other half find it on the left side of the brain. And actually, these functions are not associated with specific parts of the brain. Just because blood is flowing in these imaging studies does not mean that those parts of the brain are necessarily responsible for emotion. And in fact, emotional functioning, like many other aspects of cognitive functioning, is occurring with the involvement of many parts of the brain at the same time. So how do we move forward? We're going to need to clean up the current mess that is cognitive neuroscience, and the article by Yarkoni and colleagues describes how we can do that. We can improve cognitive neuroscience by making the data and images widely available for reanalysis and synthesis. That's really important. We have to fix the flawed data analyses. At this point, scholars have estimated that at least half of the published articles in cognitive neuroscience in the top journals are fatally flawed and need to be reanalyzed. So there's a lot of work to go back and fix that. And then we need to conduct meta-analyses of data from multiple studies. This is going to take some time. And meanwhile, what do we know? What can we conclude about cognitive neuroscience? Our current understandings of essential patterns of brain development probably won't change. So there are some basic ideas here that we're pretty confident in. So these ideas are that there is a hierarchical pattern of development in the brain from simpler to more complex functions. Simpler functions are established early, more complex functions established later. The brain is experience dependent, meaning that the experiences that we have shape how our brains evolve, particularly in relation to language. The brain is use dependent. If we don't use certain functions, we will lose them. The pattern of rapid development in the early years seems well established. The idea that there are sensitive periods for some kinds of functions to develop, also well established. This pattern of scaffolding, building structures, equifinality, multifinality, and plasticity, these ideas are pretty well established, particularly the idea that plasticity decreases over time. However, some current hypotheses about functions of specific regions of the brain may not hold up. For example, the ideas about right and left hemispheres look like they will not hold up. So what are the implications for social work? All of this suggests that early childhood development is very important because the brain is developing so rapidly and is so plastic in this time that the events of early childhood really do matter. And also early intervention is most effective. So prevention and intervention are important. Again, we want to reduce exposure to risk factors and enhance access to protective factors. This suggests that adequate nutrition, safe housing, economic resources, and parent support are all very important in the early years. It's important to conduct screening and early intervention for developmental problems, and it's important to treat traumatic brain injury as quickly as possible. Here's some further references for you.